Chapter 1, Cellular Adaptations, Cell Injury, and Cell Death. Introduction to Pathology. Pathology is literally the study, logos, of suffering, pathos. More specifically, it is a bridging discipline involving both basic science and clinical practice and is devoted to the study of the structural and functional changes in cells, tissues, and organs that underlie disease. By the use of molecular, microbiologic, immunologic, and morphologic techniques, pathology attempts to explain the whys and wherefores of the signs and symptoms manifested by patients while providing a sound foundation for rational clinical care and therapy. Traditionally, the study of pathology is divided into general pathology and special, or systemic, pathology. The former is concerned with the basic reactions of cells and tissues to abnormal stimuli that underlie all diseases. The latter examines the specific responses of specialized organs and tissues to more or less well-defined stimuli. In this book, we first cover the principles of general pathology and then proceed to specific disease processes as they affect particular organs or systems. The four aspects of a disease process that form the core of pathology are its cause, etiology, the mechanisms of its development, pathogenesis, the structural alterations induced in the cells and organs of the body, morphologic changes, and the functional consequences of the morphologic changes, clinical significance. Etiology or cause. The concept that certain abnormal symptoms or diseases are caused is as ancient as recorded history. For the Akkadians, 2500 BC, if someone became ill, it was the patient's own fault, for having sinned, or the makings of outside agents, such as bad smells, cold, evil spirits, or gods. 1. In modern terms, there are two major classes of etiologic factors, intrinsic or genetic, and acquired, for example, infectious, nutritional, chemical, physical. The concept, however, of one etiologic agent for one disease, developed from the study of infections or single-gene disorders, is no longer sufficient. Genetic factors are clearly involved in some of the common environmentally induced maladies, such as atherosclerosis and cancer, and the environment may also have profound influences on certain genetic diseases. Knowledge or discovery of the primary cause remains the backbone on which a diagnosis can be made, a disease understood, or a treatment developed. Pathogenesis. Pathogenesis refers to the sequence of events in the response of cells or tissues to the etiologic agent, from the initial stimulus to the ultimate expression of the disease. The study of pathogenesis remains one of the main domains of pathology. Even when the initial infectious or molecular cause is known, it is many steps removed from the expression of the disease. For example, to understand cystic fibrosis is to know not only the defective gene and gene product, but also the biochemical, immunologic, and morphologic events leading to the formation of cysts and fibrosis in the lung, pancreas, and other organs. Indeed, as we shall see throughout the book, the molecular revolution has already identified mutant genes underlying a great number of diseases, and the entire human genome has been mapped. Nevertheless, the functions of the encoded proteins and how mutations induce disease are often still obscure. Because of technological advances, it is becoming increasingly feasible to link specific molecular abnormalities to disease manifestations and to use this knowledge to design new therapeutic approaches. For these reasons, the study of pathogenesis has never been more exciting scientifically or more relevant to medicine. Morphologic changes. The morphologic changes refer to the structural alterations in cells or tissues that are either characteristic of the disease or diagnostic of the etiologic process. The practice of diagnostic pathology is devoted to identifying the nature and progression of disease by studying morphologic changes in tissues and chemical alterations in patients. More recently, the limitations of morphology for diagnosing diseases have become increasingly evident, and the field of diagnostic pathology has expanded to encompass molecular biologic and immunologic approaches for analyzing disease states. Nowhere is this more striking than in the study of tumors. Breast cancers and tumors of lymphocytes that look morphologically identical may have widely different courses, therapeutic responses, and prognosis. Molecular analysis by techniques such as DNA microarrays has begun to reveal genetic differences that bear on the behavior of the tumors. Increasingly, such techniques are being used to extend and even supplant traditional morphologic methods. Functional derangements and clinical manifestations. The nature of the morphologic changes and their distribution in different organs or tissues influence normal function and determine the clinical features, symptoms and signs, course, and prognosis of the disease. Virtually all forms of organ injury start with molecular or structural alterations in cells, a concept first put forth in the 19th century by Rudolf Virchow, known as the father of modern pathology. We therefore begin our consideration of pathology with the study of the origins, molecular mechanisms, and structural changes of cell injury. Yet different cells in tissues constantly interact with each other, and an elaborate system of extracellular matrix is necessary for the integrity of organs. Cell-cell and cell matrix interactions contribute significantly to the response to injury, leading collectively to tissue and organ injury, which are as important as cell injury in defining the morphologic and clinical patterns of disease. Overview: cellular responses to stress and noxious stimuli. The normal cell is confined to a fairly narrow range of function and structure by its genetic programs of metabolism, differentiation, and specialization, by constraints of neighboring cells, and by the availability of metabolic substrates. It is nevertheless able to handle normal physiologic demands, maintaining a steady state called homeostasis. More severe physiologic stresses and some pathologic stimuli may bring about a number of physiologic and morphologic cellular adaptations, during which new but altered steady states are achieved, preserving the viability of the cell and modulating its function as it responds to such stimuli. Figure 1.1 and Table 1.1. The adaptive response may consist of an increase in the number of cells, called hyperplasia, or an increase in the sizes of individual cells, called hypertrophy. Conversely, atrophy is an adaptive response in which there is a decrease in the size and function of cells. It also occurs in certain pathologic conditions, when cells are damaged beyond repair, and especially if the damage affects the cell's nuclear DNA. We will return to a detailed discussion of these pathways of cell death later in the chapter. Stresses of different types may induce changes in cells and tissues other than adaptations, cell injury, and death. See Table 1.1. Cells that are exposed to sublethal or chronic stimuli may not be damaged but may show a variety of subcellular alterations. Metabolic derangements in cells may be associated with intracellular accumulations of a number of substances, including proteins, lipids, and carbohydrates. Calcium is often deposited at sites of cell death, resulting in pathologic calcification. Finally, cell aging is also accompanied by characteristic morphologic and functional changes. In this chapter, we discuss first how cells adapt to stresses, and then the causes, mechanisms, and consequences of the various forms of acute cell damage, including cell injury and cell death. We conclude with subcellular alterations induced by sublethal stimuli, intracellular accumulations, pathologic calcification, and cell aging. Cellular adaptations of growth and differentiation. Cells respond to increased demand and external stimulation by hyperplasia or hypertrophy, and they respond to reduced supply of nutrients and growth factors by atrophy. In some situations, cells change from one type to another, a process called metaplasia. There are numerous molecular mechanisms for cellular adaptations. Some adaptations are induced by direct stimulation of cells by factors produced by the responding cells themselves or by other cells in the environment. Others are due to activation of various cell surface receptors and downstream signaling pathways. Adaptations may be associated with the induction of new protein synthesis by the target cells, as in the response of muscle cells to increased physical demand, and the induction of cellular proliferation, as in responses of the endometrium to estrogens. Adaptations can also involve a switch by cells from producing one type of proteins to another or markedly overproducing one protein, such is the case in cells producing various types of collagens and extracellular matrix proteins in chronic inflammation and fibrosis, Chapter 2 and Chapter 3. Figure 1-2 The relationships between normal, adapted, reversibly injured, and dead myocardial cells. The cellular adaptation depicted here is hypertrophy, and the type of cell death is ischemic necrosis. In reversibly injured myocardium, generally effects are only functional, without any readily apparent gross or even microscopic changes. In the example of myocardial hypertrophy, the left ventricular wall is more than 2 cm in thickness, normal is 1 to 1.5 cm. In the specimen showing necrosis, the transmural light area in the posterolateral left ventricle represents an acute myocardial infarction. All 3 transverse sections have been stained with triphenyltrazolium chloride, an enzyme substrate that colors viable myocardium magenta. Failure to stain is due to enzyme leakage after cell death. Figure 1-3 Physiologic hypertrophy of the uterus during pregnancy. A. Gross appearance of a normal uterus, right, and a gravid uterus, removed for postpartum bleeding, left. B. Small spindle-shaped uterine smooth muscle cells from a normal uterus, left, compared with large plump cells in gravid uterus, right. Figure 1-4 Changes in the expression of selected genes and proteins during myocardial hypertrophy. Figure 1-5 A. Atrophy of the brain.